Now let's learn some topics related to molecular biology. So before moving on to the complex topics, let's learn what is molecular biology. Molecular biology is the branch of biology that deals with the study of biological activity at the molecular level. It involves the study of the structure and function of macromolecules such as DNA and RNA and proteins and how they interact within and between cells. Here are some of the important advantages, disadvantages and applications of molecular biology. Importance: understanding of life processes. Molecular biology has allowed scientists to understand the intricate processes that govern life, including gene expression, DNA replication, and protein synthesis. Advancement in medicine. Molecular biology has contributed to the development of various medical applications, including diagnostic tests, vaccines, and gene therapies. Then, development of biotechnology. Molecular biology has been instrumental in the development of biotechnology, which has allowed for the production of novel drugs, food products, and industrial chemicals. Then advantages include precision and accuracy. Molecular biology techniques are highly specific and precise, allowing for the detection of even small amounts of genetic material. Then speed. Many molecular biology techniques are rapid, allowing for the analysis of large numbers of samples in a short period. Then versatility. Molecular biology techniques can be applied to a wide range of biological systems and samples, from bacteria to human tissues. Disadvantages include cost. Many molecular biology techniques require expensive equipment and reagents, which can be a barrier to their use. Then complexity. Molecular biology techniques often involve complex protocols and require skilled technicians to perform. Then ethical concerns. Molecular biology techniques, particularly those involving genetic engineering, raise ethical concerns regarding their potential impact on society and the environment. Applications: disease diagnosis. Molecular biology techniques are used for the detection of pathogens and genetic mutations associated with diseases. Then forensics. Molecular biology techniques are used in forensic investigations to identify suspects and victims and to establish the presence of biological material in a crime scene. Then genetic engineering. Molecular biology techniques are used to modify the genetic material of organisms for research or commercial purposes, including the production of genetically modified crops and animals. So in conclusion, molecular biology has had a profound impact on our understanding of life processes, and its applications in medicine, biotechnology, and forensics have revolutionized these fields. However, the use of molecular biology techniques also raises ethical concerns, and their cost and complexity can be a barrier to their widespread use. First topic: semi-discontinuous synthesis. Okazaki fragments. Semi-discontinuous DNA synthesis is a mechanism by which the lagging strand of DNA is synthesized during DNA replication. The synthesis of the lagging strand is discontinuous because it is synthesized in short fragments called Okazaki fragments. Here is a detailed explanation of this topic. The lagging strand of DNA is synthesized in the opposite direction of the replication fork movement. Therefore, the synthesis of the lagging strand occurs in small fragments of DNA ranging from 1000 to 2000 nucleotides in length. These fragments are called Okazaki fragments and are synthesized in the 5' to 3' direction. In the 5' to 3' direction. Never forget that direction. Then let's learn about the mechanism of this semi-discontinuous DNA synthesis mechanism. The semi-discontinuous DNA synthesis mechanism involves 3 steps. First, initiation, next, elongation, and termination. The first process is the initiation. We know that the initiation of DNA synthesis on the lagging strand occurs after the initiation of DNA synthesis on the lagging strand occurs after the primase enzyme synthesizes a short RNA primer. The RNA primer provides a free 3-OH group for DNA polymerase III to add nucleotides in the 5' to 3' direction. The leading strand is synthesized continuously in the same direction as the replication fork movement. elongation. After the RNA primer is synthesized, DNA polymerase III binds to the primer template junction and begins to synthesize the first Okazaki fragment. As DNA polymerase III adds nucleotides to the 3' OH end of the RNA primer, it displaces the RNA primer, creating a gap in the newly synthesized DNA strand. The RNA primer is then removed by the RNase H enzyme, and the resulting gap is filled in by DNA polymerase I using the 3' OH end of the adjacent Okazaki fragment as a primer. This process of gap filling and Okazaki fragment synthesis continues until the lagging strand is fully synthesized. Then termination process. Once the lagging strand is fully synthesized, the gaps between Okazaki fragments are sealed by DNA ligases, which catalyzes the formation of a phosphodiester bond between adjacent nucleotides. Major important points. Semi-discontinuous DNA synthesis occurs on the lagging strand of DNA during replication. The lagging strand is synthesized in short fragments called Okazaki fragments. The synthesis of the leading strand is continuous in the 5' to 3' direction. The initiation of DNA synthesis on the lagging strand requires the synthesis of an RNA primer by primase. The synthesis of the Okazaki fragments requires the coordinated action of RNA polymerase III, RNase H, and DNA polymerase I. The gaps between Okazaki fragments are sealed by DNA ligase. In conclusion, semi-discontinuous RNA synthesis is a crucial mechanism in the DNA replication that allows for the efficient and accurate synthesis of both the leading and lagging strands of DNA. The synthesis of Okazaki fragments is essential to ensure the continuity of the lagging strand and is a result of the opposite direction of DNA synthesis on the lagging strand compared to the leading strand. Next topic, replication origin and replication fork in detail. So replication origin and replication fork are two critical components of the DNA replication process. Replication origin. The replication origin is a specific sequence of nucleotide in DNA where DNA replication begins. It is recognized by the replication initiation proteins that bind to the origin and recruit the DNA helicase enzyme to initiate DNA replication. In bacteria, the replication origin is a single site on the circular chromosome. In eukaryotes, the replication origin is much more complex and consists of multiple sites that are recognized by different proteins. The replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery. For example, in bacteria, the replication origin is called ORI-C, and it is a 245 BP sequence located at a specific site on the circular chromosome. In humans, the replication origin is a complex sequence that consists of multiple sites located throughout the genome. The replication origin in humans is essential for the initiation of DNA replication and the maintenance of genome stability. Now, what is the importance? The replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery. The recognition of origin by replication initiation proteins ensures that replication occurs in a controlled and regulated manner. The replication origin also plays a critical role in maintaining genome stability by ensuring that each chromosome is replicated once and only once during each cell division cycle. Then, replication fork. The replication fork is the site where DNA replication occurs. It is formed by the unwinding of the double helix by the DNA helicase enzyme, which separates the two strands of DNA. The replication fork is composed of two strands, the leading strand and the lagging strand. The leading strand is synthesized continuously in the 5' to 3' direction, whereas the lagging strand is synthesized in short fragments called Okazaki fragments in a semi-discontinuous manner. The replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. In bacteria, the replication fork is a single site on the circular chromosome where DNA replication occurs. In eukaryotes, the replication fork is much more complex and consists of multiple sites where DNA replication occurs simultaneously. The replication fork in eukaryotes is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. So what is the importance? The replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. The leading and lagging strands are synthesized simultaneously at the replication fork, allowing for the efficient duplication of the genome. The replication fork also play a critical role in maintaining genome stability by preventing the accumulation of DNA damage and ensuring the faithful transmission of genetic information to daughter cells. In conclusion, replication origin and replication fork are two critical components of the DNA replication process. The replication origin is essential for the initiation of DNA replication and the coordination of the replication machinery, while the replication fork is essential for the efficient and accurate synthesis of new DNA strands during DNA replication. Next, understanding structure and function of the replication origin and replication fork is essential for understanding the process of DNA replication and the maintenance of genome stability. And the replication fork also play a critical role in maintaining genome stability by preventing the accumulation of DNA damage and ensuring the faithful transmission of genetic information to daughter cells. Next topic, unit of replication, extrachromosomal replication of bacteria, TI plasmid. Then unit of replication and extrachromosomal replicon are two important concepts related to bacterial genetics. The unit of replication. The unit of replication is the segment of DNA that is replicated from a single origin of replication. In bacteria, the unit of replication is often referred to as replicon, and it includes the DNA sequence that is replicated from a single origin of replication to the point where the replication machinery meets at the opposite end of the replicon. The size of the replicon can vary depending on the organism, and it can range from a few thousand base pairs to millions of base pairs. For example, in bacteria, the unit of replication can be a plasmid or a segment of chromosome. In the case of Escherichia coli bacterium, the chromosome has multiple origins of replications, and each of these origins represents a unit of replication. Additionally, bacteria plasmids such as F plasmid also have a single origin of replication that serves as the unit of replication. Then what is the importance? The concept of unit of replication is important in the study of bacterial genetics as it helps us understand how bacterial DNA is replicated. By identifying the origin of replication and the direction of replication, we can study the replication process in more detail and gain insights into the mechanisms that govern DNA replication. Then extrachromosomal replicon of bacterial TI plasmid. The bacterial TI plasmid is a large circular DNA molecule that is found in Agrobacterium tumefaciens. This plasmid is an extrachromosomal replicant that allows it to replicate independently of the bacterial chromosome. The extrachromosomal replicon of the TI plasmid is known as the virulence region, and it contains all the genes required for the transfer of plasmid into the plant host. Then procedure. The extrachromosomal replicon of the TI plasmid is replicated in a similar manner to the chromosomal DNA. The replication process is initiated at a specific site on the plasmid known as the origin of replication, and the plasmid is replicated bidirectionally from the site. The replicon is then partitioned into the daughter cells during cell division, allowing for the stable maintenance of the plasmid within the bacterial population. For example, the TI plasmid is commonly used as a vector in genetic engineering as it can transfer genes into plant cells. By inserting genes of interest into the virulence region of the plasmid, researchers can create genetically modified plants that express new traits such as resistance to herbicides or increased yield. What is importance? The extrachromosomal replicon of the TI plasmid is important in genetic engineering as it allows for the stable maintenance of the plasmid within bacterial populations. This makes it an attractive tool for the transfer of genes into plant cells, allowing for the creation of genetically modified plants with new traits. Additionally, the study of the TI plasmid has helped us understand the mechanisms that govern bacterial plasmid replication and the transfer of DNA between bacterial cells. Then in conclusion, the unit of replication and extrachromosomal replicon are two important concepts in bacterial genetics. The unit of replication is the segment of DNA that is replicated from a single origin of replication, while the extrachromosomal replicon is the DNA segment that allows a plasmid to replicate independently of the bacterial chromosome. Understanding these concepts is important in the study of bacterial genetics and the development of genetic engineering tools. Then enzymes or proteins of replication. Enzymes and proteins are key players in the process of DNA replication. Here is a detailed explanation of the major enzymes and proteins involved in replication. DNA helicase. DNA helicase is a protein that unwinds the double helix structure of the DNA molecule. The enzyme breaks the hydrogen bonds that hold the two strands together, allowing the DNA strands to separate and form a replication fork. For example, E. coli DNA helicase is known as DNAB, while the human DNA helicase is known as MCM. In the sense, mini chromosome maintenance protein. What is importance? DNA helicase plays a crucial role in the initiation of DNA replication. Without this enzyme, the DNA strands would remain stuck together and replication could not occur. Then DNA polymerase is an enzyme that adds nucleotides to the growing DNA chain. The enzyme can only add nucleotides in the 5' to 3' direction, meaning that it can only add nucleotides to the free 3' end of the DNA chain. For example, E. coli DNA polymerase is known as DNA pol 3, while the human DNA polymerase is known as, I'm not sure about that, I'm sorry. Then importance. DNA polymerase is essential for the replication of DNA. It adds nucleotides to the growing chain, ensuring that the new DNA strand is a faithful copy of the original DNA strand. Then DNA ligases. DNA ligase is an enzyme that catalyzes the formation of a phosphodiester bond between adjacent nucleotides in the DNA strand. The enzyme is responsible for sealing the gaps in the lagging strand of the DNA molecule where the Okazaki fragments have been synthesized. So what is DNA ligase? This is used to catalyze the formation of phosphodiester bond between adjacent nucleotides in DNA strand. Then what is the importance of this? DNA ligase is important for the completion of DNA replication. Without this enzyme, the Okazaki fragments in the lagging strand would remain unconnected and the DNA strand would be incomplete. Then single-stranded DNA binding proteins. Single-stranded DNA binding proteins are proteins that bind to the single-stranded DNA that is formed during the replication process. The proteins prevent the DNA strands from reannealing, ensuring that the DNA remains in its single-stranded state. An example, E. coli single-stranded DNA binding protein is known as SSB, while the human single-stranded DNA binding protein is known as RPA, in the sense replication protein A. What is importance? Single-stranded DNA binding proteins play a critical role in the replication process. They prevent the DNA strands from reannealing, which would disrupt the replication process. Then primase. Primase is there. Primase is an enzyme that synthesizes short RNA primers on the lagging strand of the DNA molecule. The RNA primers provide a free 3' end of the DNA polymerase to start synthesizing the Okazaki fragments. For example, E. coli primase is known as DNAG, while the human primase is known as PRIM1 and PRIM2. What is importance? primase is important for the initiation of DNA synthesis on the lagging strand. Without the RNA primers, the DNA polymerase would have no free 3' end to start synthesizing the Okazaki fragments. Thank you for listening.